Welcome to Life and Biology. I'm Dr. Joel Graff, and today we're going to talk about laying down Mendel's laws. So there's two laws that we're going to discuss that Mendel was able to come up with after he did 8 years worth of plant experiments with pea plants. And I've heard a story that Mendel was originally going to do his experiment with mice, but he was a monk, and having mice have sex at the monastery was a bit of a push, a little too risque, and so then he had to have plants have sex. And so when we're talking about Mendel's genetics, we're talking about organisms that go through sexual reproduction. Okay, so let's say this is a cell of an organism that's going to go through reproduction, and there's two types of reproduction. We can do mitosis, so that you end up with two daughter cells that had the same information as the parent cell, or we can go through meiosis, and this is meiosis 1, which is the first replication cycle, and then there's meiosis 2, and that's when you end up with gametes. So what I want to focus on with this slide is that you've got two chromosomes, one type of chromosome, but two chromosomes, a red chromosome and a blue chromosome. These are homologous chromosomes. If we looked at the genes that were on each of these chromosomes, they would line up similarly on those two. So this is a cell that has one pair of homologous chromosomes. If you go through S phase, and you go through S phase for both mitosis or meiosis, doesn't matter, you're going to, meiosis 1, you're going to end up with a couple of X's. So there's where dos Equis comes from. So if you have these two X's here, they're sister chromatids. The blue chromosomes are copies of each other. The red chromosomes are copies of each other. So those are called sister chromatids. If we were to go through mitosis, those X's line up along the equator of the cell. If you consider the centromeres set up at either end of the cell as the poles of the cell, then you've got those X's lined up along the equator. And then if you look at meiosis 1, the X's are not lined up on top of each other, but they're lined up side by side. Now think this through. What will happen if those microtubules start getting pulled and you separate out your sister chromatids and the cell divides down the center? Well, you're going to get a blue chromosome copy going towards that cell, and a blue chromosome copy going towards this cell, and a red chromosome copy going towards that cell, and a red chromosome copy going towards this cell. So that when the cell does divide down the middle there, each of the daughter cells will end up with one red chromosome and one blue chromosome, which is exactly what we started with. Now, things are a little bit different when you go through meiosis because they're side by side. When they get pulled apart, one cell will end up with two copies of the red chromosome, sister chromatids of the red chromosome, and the other cell will end up with both copies of the blue chromosome. So that brings us to Mendel's first law of segregation. Now remember, Mendel didn't know about the genetics, but he was able to come up with these laws based on all of these genetic crossings that he did with the pea plants. So, law of segregation. We're gonna start with a cell here that has, in this case, we've got a green chromosome and a purple chromosome. And I'm sorry, the colors kind of wash out, but and on the green chromosome, let's say that came from the maternal side of this organism, and the purple chromosome came from the maternal side. So we got paternal dad's side, maternal mom's side, and we have these letters here, capital A and lowercase a with a dot. What I'm doing there is I'm indicating the type or the allele, the version of the gene that you would find on the green or the purple chromosome. Now there's lots and lots of genes on each of these chromosomes. We're just focusing in on this particular gene at this particular spot in the chromosome a particular place in the chromosome is called a locus. So at this locus, there's a chromosome that encodes the capital A version of the gene. And on this locus, we have the lowercase a version of the gene. And because they have different letters, it means that they have slightly different DNA sequence. And so this could be things like the color of the petals on a flower of the pea plants. One might be designated lowercase a and one might be designated uppercase a for purple or white. Excuse me. All right, so that's the setup. We've got this one cell with a pair of homologous chromosomes with a different allele. So it has two different types of alleles within that one cell. During meiosis 1, that's the first round of splitting of cells, we go to the stage of metaphase, and that's when the X's get hooked up to the sister chromatids get hooked up to the microtubules, because you're going to split this cell left and right. It's going to separate on the left and right and go towards each pole of the cell. And we've got the green chromosome sister chromatids on the left side, and we've got the purple sister chromatids over here on the right side of the screen. And then you pull those apart like we discussed before, so that one of the daughter cells gets the sister chromatids that are green, the other daughter cell gets the sister chromatids that are purple. And remember, the green had a capital A version of a gene or allele, and then we have a lowercase a version or allele of the gene here. And then when those get to meiosis of the next of meiosis 2, they actually look a lot like this. And then you pull those apart, and so then you end up with one cell that has just one chromosome. The sister chromatids got pulled apart, and you have the lowercase a. Again, you have the lowercase a. And then you have two cells that have the uppercase a, and they have the green version of the chromosome. Those two had the purple version of the chromosome. Okay, so that's the law of segregation. So if this organism, if these were gametes of this organism, these gametes would go find another gamete from another organism of the same species. They could be fertilized together so that you end up with a new diploid organism. Okay, so that's the law of segregation. Says that the information that an organism gets from its mother and an information an organism gets from its father gets split up at the time of when that organism makes its gametes. So that law is fairly straightforward, but we're going to lay down the next law, and the next law takes that idea and adds to it, and we're going to start to begin to see just how complex or how much mixing and matching we can do with sexual reproduction. Okay, so I've got two different cells to start with here. We've got a cell that has a blue and red chromosome that are long. We're going to call that chromosome 1. And it also has a purple and a green chromosome. And so this is two pairs of homologous chromosomes. Chromosome 1 is the longer one, chromosome 2 is the shorter one. Oftentimes the numbering system goes with whatever's longest gets the number 1, and whatever's the next longest gets the number 2, and so on. Some species, it's flipped around, so there's not always consistency in biology. But we had blue on the left and red on the right, purple on the left and green on the right here. And this is like meiosis 1. And then let's say we were to separate those out so that the red and the green would end up in one of the daughter cells, and the purple and the blue would end up in the other daughter cell. So that could happen. But let's take a look at this cell. In this case, during meiosis 1, you've got the blue chromosome on the left and the red chromosome on the right, which was what we had up here. And then we have the green chromosome on the left and the purple chromosome on the right, which is backwards. So that means that each pair of homologous chromosomes that line up during meiosis 1 they can be independent of each other. Just because the blue's on the left up here and the red's on the right doesn't mean that the green and purple one have to be in any particular order. They can be independent. So this is independent of this. So there's where our word independent comes from. Okay, so then if that were to split and you go to meiosis, metaphase of meiosis 2, you would end up with a cell that has the blue sister chromatids and green sister chromatids, red and purple. So blue and green, red and purple. But up here, it was red and green and blue and purple because the bottom chromosome pair, chromosome number 2, was lined up in the other direction. So if you just follow through on those colors, and I really hope this shows up better on your computer screen than it's showing up in just the reflection of my camera. But then you go from meiosis to metaphase of meiosis 2, and you split those cells out, then you have a cell here that has a red and a green chromosome, and another cell that has a red and green. Here's a cell that has a blue and purple. Trust me, they're blue and purple. Here we've got a blue and a green, a red and a purple, blue and green, red and purple. So we have 4 types of gametes there. So what I'm getting at is that we started out with 2 chromosomes, and then we go through meiosis 1, meiosis 2, and we end up with 4 different varieties of gametes that you could have. Or 2 different here. So that doesn't seem like that's that much big of a deal, but let's say if there was 3 chromosomes, I would in no way have the ability to write out all the possibilities on this slide. If we had 3 chromosomes, then they could mix and match in a larger number of ways. And so we go from, if you have 1 chromosome, there's only 2 to the 1 type of gametes that you can end up with, which is what we had here. So there's 2 types of gametes there. Over here, we have 2 chromosomes, and 2 to the 2 is 4. So there are 4 different types of gametes that we ended up with over here. Now, if we had 3 chromosomes, it would be 2 to the 3, which is 8. And so you could end up with 8 different types of gametes. So each organism that has 3 chromosomes could mix and match and come up with 8 different types of gametes. And then they could mate with someone that has 3 chromosomes, and then they mix and match their 8. And so then you can take the 2 different versions of 8 possible chromosome pairs, and you've got a lot of different opportunities for mixing and matching there. Now let's go up to 4 chromosomes. That's 2 to the 4. So we're up to now 16 possibilities for your gametes as far as which combination of chromosomes they got based on the maternal and paternal chromosomes that the organism got from its parents. And you can see that genetics is hard to talk about once you start getting into it, but that would give you 16 different versions. And so then when you have that organism with 4 chromosomes that has 16 possible gametes mate with another one, they could have lots of babies together, and none of those babies would likely ever be identical to each other because they're mixing and matching. Now, humans have 23 chromosomes, and so you have to take 2 to the 23rd, which I guess you're going to have to pull out your calculator for that one because I'm not going to be able to do it. But you can see that if a male or female produces sperm and eggs, that you're not going to have any identical sperm and eggs with so many possibilities going back and forth between all those chromosomes. Just following those 2 laws, the law of segregation and the law of independent assortment, gives you an almost unlimited number of possible gametes, sperm or egg, that a person could make, depending on which sex they are. So The next time we talk, we're going to actually bring in one more topic, and that's crossing over or recombination. And that will just take those numbers that we just came up with all the independent assortment and law of segregation and law of independent assortment, and it will basically multiply that to infinity the type of different sperm or eggs you could come up with from all that. So two fairly simple concepts, and usually just drawing it out on a board like I had, it's you can wrap your head around it. But once you get up to more than, say, 3 chromosomes, it gets really confusing. And so that's why there's a lot of organisms that do sexual reproduction that have multiple chromosomes, and in that way they can have unlimited mixing and matching, and so that the generations are different. I'll go ahead and stop there because this video is getting a little longer than I like them to be. But anyway, know those 2 laws that Mendel learned just from his pea plant experiments. Talk to you later.